Now the next one organelle, which is peroxisome. Okay, so peroxisome is basically what? They are the metabolic compartment bound by a single membrane. Okay, you have to mention, remember, it is a single membrane bound again organelle. The enzymes, they have certain enzymes. Okay, so what is basically function of peroxisome? It remove the hydrogen atom from certain molecules and transfer it to O2 and make the H2O2. Okay, what is saying, whatever the enzyme is present over here, what it do, it remove the hydrogen from any material and mix it with what? Hydrogen plus O2, and they make the H2O2. This H2O2 is called the hydrogen peroxide. Okay, so what basically it is doing, its main function is detoxify alcohol and harmful compounds in the liver. See, if there's any toxic substance or some alcohol that we use to consume, it goes directly into our stomach. From stomach it enter into our liver. So here in liver, we actually the main function of liver to detoxification, whatever the bad things or some toxic things which are enter into our body, the main function of liver is to detoxify it. Okay, how it detoxifies? Yeah, so liver contain large amount of peroxisome. Okay, so what it peroxisome do, it takes the hydrogen ion from these alcohol harmful compounds. It takes the hydrogen ion and mix it with O2, and ultimately it make the H2O2. Got it? Okay, so this is how it just remove the toxic substances from these compounds. Got it? Though H2O2 is also very toxic, it is also toxic. It's not like that it is not toxic, but there are certain other enzymes, or you can say degraded by other enzymes. Okay, there are certain other enzymes in the body which further degrade this H2O2, remove in the form of water content. How? See, other enzyme degrade it and convert it into water. Okay, plus certain amount of oxygen. So this is how the toxic substance is removed from the body. Got it? So the next point is what? They grow larger by incorporating protein and lipid. Okay, because they are surrounded by a membrane, and we know very well that membrane composition is what? Protein and lipid. Okay, membrane composition is protein and lipid. So they takes up the protein and lipid and just increase its size and detoxify the molecules continuously. Got it? Let's go further. Next organelle is cytoskeleton. It's cytoskeleton. If in a very simple language I need to tell you, suppose this is the cell. So what is cytoskeleton? They are the some fibers, and there's a network of fiber like this. Okay, in the whole cell it just spread. Okay, it's like a you can consider it like a pillar. So many pillars in a room, they are working like this. So it's a network of fiber that organizes the structure and activities of the cell. So what is actually doing? It maintain the shape of the cell. Okay, because they acting like a pillar, so many pillars in a room, so they just maintain all the walls, all the cell structure. So let's go first function. So first is function is mechanical support. So definitely if they act like a pillar, they give the support, they maintain the shape as well. Okay, suppose this is the cell, right? If there's no this fiber, the cell will collapse. There's a chances that it will collapse. So it maintain the shape of the cell because of these fibers. Next is what? They provide the anchorage for organelles. See, now there's different organelles are present inside this whole cell. These are what? Organelles. So these organelles will not go to their other places. I mean, it cannot shift over here. This cannot go to other place. They are kept in their place only with the help of these cytoskeleton. So it provides anchorage for the organelles and cytosolic enzymes molecule. So they are at their space only. They do not move here and there and do not change their location in the cell. Okay. Yeah. Next point is what? Cell motility. So it provide the motility as well. Next is what? Cell motility. Motility means what? It provide the movement, you can say. It can move with the help of this cytoskeleton. Okay, so interaction of cytoskeleton with motor protein. Okay, so see, now next question arises, how it provide motility. I mean, like, how it is possible with the help of cytoskeleton the cell is able to move from one place to another. So what happen actually, there are certain proteins which are called motor protein. These motor protein actually, they show the movement, they have the energy, so that they can show the movement. So what happened, these cytoskeleton, they just interact with these motor protein, they work together, and then they work together with the plasma membrane, and this is how it moves. Let me show you how it moves. Suppose there is a vesicles. Okay, so there is a vesicles which is having something, whatever the things is present in this vesicle, it attach with the motor protein. This is the motor protein, you can see over here, this red color, it is the motor protein. Okay, now after making this complex, now it move on this cytoskeleton. See, this is the cytoskeleton, it is a fiber. Okay, it is a fiber, so it provide a track. Okay, so it acts as a track, and these motor protein move on this track, so on. Okay, suppose there's a cell, there's a one organelle over here, another is over here. Now something is being released from this one, or you just consider this is the nucleus, here is the what, ER. Okay, ER releases some or made some protein. Okay, now this protein need to enter inside this Golgi apparatus, right? But how it does know that it has to go over here only? Okay, I mean, like, it can I mean, like, it can go this one also, it can go anywhere else also. So what is there, there are certain tracks over here. These tracks are made by the cytoskeleton. On these track only these molecules run on these track, and that's why it enter or go to their right place. Okay, go to their right location with the help of these track. So here this cytoskeleton act as a track. Okay, on which these motor protein, I mean, like, the protein which is being released over here, it associates with some motor protein. What is the role of motor protein? It provides the energy for the movement. See, ATP is being given for the movement. So this is how it these molecule go along with this track and go for one location to another one. So did you get it? What is exactly cytoskeleton is providing? Yeah, it providing a track, okay, on which the things or molecules can move from one places to another. Okay, now the so next point is this one only, it provide the track. Okay, so what are the example? I mean, like, so first example, like, there are certain vesicle which contains the neurotransmitter. Okay, so I know this is a new word for you, neurotransmitter. Just for now you just understand neurotransmitter, they are certain molecules which are used in nervous system, okay, just understand right now this much only. When we have the nervous system, then I will let you know about this one detail. So these are certain chemicals, actually these are certain chemicals which are used in nervous system. So these chemicals move from one place to another. Okay, see, if I'll tell you a little bit about nerves. Okay, so if I'll say see for a nerve cell, so basically nerve cells structure is like this one. Okay, we'll do this one in detail as well in our later syllabus, later chapters. Okay, so see, this is the nerve structure like this one. Okay, so what happened, certain molecules, they move certain information, or you can say certain molecule, they move from one place to another. So this is what it is called, exon. Okay, it is called exon. Now these molecules travel from this exon and reach ultimate this location. Okay, did you get it? Reach ultimate at the tip of exon. What is this? Tip. Okay, so it ultimately reach over here. So I mean, like, how I mean, like, these molecules get to know they have to run along this one direction only. This is because of the cytoskeleton, because cytoskeleton is providing a track on which it is moving. So that is what it is telling, this vesicle or neurotransmitter migrate to tips of the exon with the help of cytoskeleton. Second example is what? The vesicles from ER. Okay, as I told you just now, if any vesicle releases from the ER, it travels to the Golgi because of the help of cytoskeleton. Third is what? Formation of food vacuole, that is infolding of plasma membrane. As I told you one more time, suppose this is a cell and there are certain food particles. So what happened actually? This cell moved towards the food particle and folded, thus infolding the plasma membrane like this one. Okay, this is what? Plasma membrane. Okay, PM is plasma membrane. So there's an infolding of plasma membrane taking this food material inside in the form of food vacuole. Okay, so this infolding, taking the material inside, that is all because of the cytoskeleton as well. So this is also a type of motility. Here the cell membrane is moving inside or enfolding inside. So this is also kind of motility. See, vesicles moving from ER to Golgi, this is also kind of motility. Neurotransmitter. So this is how it play role in cell motility. Now did you get it? I mean, how it provide the motility. Yeah. Okay, so that's all about the cytoskeleton. Now further, if we'll see different components of cytoskeleton, it is basically components of cytoskeleton. Okay, so let's see. There are three main components of cytoskeleton. One is the microtubules. They are the thickest hollow tube. Okay, they are the thickest hollow tube like this one, which is made up of tubulin. Tubulin is what? It is a protein. So it is made up of tubulin. Second is microfilament. It is thinnest and made up of actin protein. It is thinnest and made up of thinnest as well as you can say solid. Okay, it is not hollow inside. And the third one is intermediate filament. Intermediate, as you can see, name suggests itself, it is having the intermediate diameter, you can say. Okay, diameter means what? Less than this microtubule, more than this microfilament. Okay, in between these two. So it is thickest, it is thinnest, and it is intermediate one. So there is a here is the comparison of all these cytoskeletal. So first one is microtubule, second microfilament, and third is the intermediate filament. Okay, structure is what? It is a hollow tube. It is a hollow tube. It is microfilament is two intertwined strand of actin. Okay, suppose what actually happened over here, there's a two strand which are very thin and they intertwined over each other like this one, like this one. Can you see? Like this one. Yeah. Okay, and third one is fibrous protein coiled into cables. It is like this one, cables. Okay, like this one, it just coil. Fine. Now let's go further. Its diameter is 25 nanometer. It is called nanometer. NM is nanometer. With 15 nanometer lumen. Lumen is what? Lumen means cavity. Okay, the space inside. That is called lumen. It is 7 nanometer and it is 8 to 12. So you can see 25 nanometer. It's a huge one. Okay, that's why it is thickest. Protein unit. Now if we'll see, microtubule is made up of tubulin protein. What is tubulin? It is a dimer. Dimer means what? There's a two molecule which is consisting of alpha tubulin and beta tubulin. Okay, so there's a two tubulin, alpha and beta. They both combine together and make a dimer. So it is always present in a dimer form. As a protein is actin, and one here is intermediate, one of the several different protein. There are so many different kind of protein. For example, keratin. For example, keratin. Okay. Next is what? Main function. Its main function is maintenance of the cell shape, cell motility, chromosome movement in cell division, and organelle movement. We will do all these things. Okay. Okay. Now next is what it do? It maintain the shape again, change in cell shape, muscles contraction, cytoplasmic streaming, and cell motility and cell division. Okay. I'll explain everything. And again here is also maintenance of the shape. So if you will see, all three are playing role in maintenance of the cell shape. Okay. It is common in all three. Again, anchorage of nucleus and certain other organelle and formation of nuclear lamina. So if you remember, nuclear lamina that was present under the nuclear membrane. Okay, I have told you if you remember. So here is microtubule is given. See, hollow tube, it is there. If we see the structure of this microtubule, it is like this. Okay, so there's a row of tubulin dimer. So there's a tubulin dimer is present and they are coiling like this. Like this. Okay, and this is how they make a hollow tube. In this one, actin tube protein, they are coiling around each other, so they make a thickest, you can say solid and thinnest tube. And here this is coil. Okay, so here the protein which is prominently present over here, it is keratin protein. Got it? Now let's take each and every one further in detail. So let's take microtubule in detail again. So as I told you, it is made up of tubulin protein which is present in dimer. So there is a alpha tubulin and beta tubulin. Okay, it is alpha tubulin is okay. So see, this is called alpha and this is called beta. Okay, so alpha tubulin and beta tubulin. Now it helps in separation of chromosomes during cell division. Now what is this? See, first of all, what do you mean by cell division? It means what? A cell is divided into their daughter cell. I mean, this is, you can consider it is a parent cell which is dividing, and the offspring which it gave rise is generally called daughter cell. Okay, so this is called cell division. Okay, now during the cell division, what actually happened? Now you must be remembering, this is the structure of a chromosome. I told you earlier when we did the nucleus. Okay, so in this one, these two are called the sister chromatid. That also I told you, sister chromatid. So what exactly sister chromatid? They are identical with each other. They are identical with each other. So what happened? This microtubule help in the separation of these sister chromatid like this one. So there's a one separation occurs, so both goes towards the opposite side like this one. So if they separated, it is like this. Okay, so these sister chromatid are get separated with the help of microtubule during the cell division. So one is goes into the one daughter cell, another one goes into another one daughter cell. Okay, so this is how division takes place. This is how division takes place, and chromosome divided into two daughter cells. Got it? Yeah. Now the next thing is what? There's one term, centrosome and centriole, and this one also I've told you, if you remember, this is only present in animal. This is only present in animal. It is not present in plant cell. Okay. The main function of centrosome and centriole is actually division. It helps in division. So what is exactly centrosome? They are made up of microtubules organizing center. I mean, like, it is a center from where so many microtubule arises near the nucleus, is always present near the nucleus. Let me revise you that one, if you remember. 1 minute. Let me show you. Yeah. So do you remember? This one is structure. This one I told you, it is centrosome, which is always present nearby the nucleus. Okay, so that one I was talking about. So let's further take this one. Okay, so centrosome, what is do? It made so many microtubule arises from this one near the nucleus. And what is actually do? These microtubule make a rope-like structure. See, microtubule, they connected together like this one, and they make a rope-like structure. So what it do? The chromosomes, what we need to separate? These chromosomes, they are sister chromatid. We need to separate that. These two sister chromatid. So what we need to separate? We need to separate like this. So these making a rope-like structure attach here. One rope is attached over here, and they both pull apart from each other in opposite direction. Because of the pulling on opposite direction, these two chromatid got separated and they goes to their respective place. So this is how it got separated. This one. Got it? So that's why it is helping in cell division. That's why. So basically here this centriole, the centriole comes over here. This is the centriole. This is also centriole. Okay, you can say centriole. This is also centriole. Okay, and the thread like structure which is made by the centriole, it is called the spindle fiber. Okay, right now it is not I mean, like, just have a look only, because we are going to discuss all these things in detail later on. Okay, so what centriole? It make a rope like structure which is called spindle fiber, and it is basically made up of microtubule. Okay, so this is how microtubule play a role in cell division. Got it? Now let's have a look of this one, centriole. So there's a one pair of centriole always in centrosome. How many centriole? Two centriole is always there. Okay, and one more thing I told you, these are always present as like a perpendicular over each other at 90-degree angle to each other. Okay, now if we'll see the structure of a centriole, it is a 9 sets of triplet microtubule. Now what does it mean? So let me show you. Okay, see what I am saying, it is always present like this one, right angle to each other. So there are two centriole over here. This is long tube like structure, and if we'll see its cross section, okay, it's simple, we just make a cross over here and make a section. If we see the cross section, there are 9 places, 1 2 3 4 5 6 7 8 and 9. At 9 places 3 microtubules are present. Can you see?
Yeah.
So 3 microtubules are present. That is what it is saying. 9 sets of triplet microtubule. This is what it is saying. Now during cell division, centriole it replicate. What happened? This centriole replicates means what? It makes its replica. It makes its one more copy. This one you just understand this much only. When we'll go in detail later on, then I'll let you know about this one. Now the thing is, what as I told you, it is only present in animal cell. Okay, but plant cell also divide, because I told you this centrosome and centriole, its main role is for division only. Now in plant cell they also divide, but centriole is absent in plant cell. Now how plant cell divide? If they do not have centriole, now how their division will take place? There is some well organized microtubules are present separately, which are dedicated for this function, for cell division. So here microtubules are absent, but this centriole is absent, but some microtubules play the role of this one. Okay, yeah, so shall we go further? Yeah, one more thing I need to tell you here. See these microtubules. So there are 9 sets are there. In center nothing is there. So they are generally say it is having a 9 plus 0 arrangement. It is just simply say 9 plus 0. 9 plus 0 means what? 9 are present at the periphery. And 0 microtubule in the center. There is no microtubule in the center. At 9 places microtubules are present on the periphery. So it is it just write like this one, 9 plus 0 arrangement. Okay, let's go further. It is next is cilia and flagella. So what is cilia and flagella? It is again consists of microtubules. Okay, see what we are doing right now. We are talking about microtubule. Now what are the different structure made by microtubule? The first one was the centrosome and centriole. Now the second structure which is made by microtubule is cilia and flagella. If you remember, cilia is what? It's like a small hair like a structure, like this one. Yeah, this is a cilia. Yeah, or long this one is structure, it is a flagella. Okay, so what it do? It consists of again microtubule, and the main role of cilia and flagella is locomotion, going from one place to another. Okay, let's talk about cilia. See where it is generally present. So it is present in trachea. Do you know trachea? Yes, it is part of your neck, or we used to say it is a part of our respiratory tract. Okay, it is a part of respiratory tract. Okay, from nostril till our lungs, that whole path is called respiratory tract, and in that one this is trachea. Okay, so in trachea what is actually there? Suppose this is a trachea. There are small hair like structure present in trachea. These hair like structure is called cilia. Okay, they are cilia. So what is role of cilia? Suppose if we are inhaling the air, if we are doing the inhalation, along with that air some dust particles comes over here. Along with air comes some dust particle comes over here. So what it do? This cilia, it is push backward, okay, out of the trachea, so that these dust particle cannot enter into our lungs. Okay, so what it do? It's work like a oars boat. That oars, what it do? They move in one direction first, then opposite direction, again one direction, then opposite direction. So they all are working synchronously in one direction, then another direction. So they work like a O's. So they move in this direction. That's why all the dust particles, they are thrown out of the trachea, so that these particles cannot enter into the lungs. So this is the role of cilia in trachea. Second one is what? It also act as a signal receiving antenna. Okay, they receive the signal from the environment. So they work like a antenna. Again, one more is reproductive tract. Okay, if I'll talk about a reproductive tract of a female reproductive tract. Okay, so let's talk about female reproductive tract. Tract means what? That pathway. So this is the female reproductive system. Okay, so here, this is the what? It is uterus. Okay, here is the ovary. These are the two ovaries which are present in a female. Okay, so from ovary, what? Some ova are released. Okay, ova, or you can say egg, are released. So this egg need to travel over here and ultimately reach into the uterus. So how it will travel? So what happened? There also certain cilia are present over on this surface. Okay, so again cilia work like a oars and push this egg forward. Okay, push this egg forward. So this is how it work again. Got it? Yeah, so this is how it work in reproductive. So I'll just write over here, pushes the egg in fallopian tube. What is this fallopian tube? It is, this whole tube is fallopian tube. I'll write over here, this is fallopian tube, or sometimes another name is oviduct also called. Okay, this one also we will talk about later. Okay, these all things are later on we will do. So in this fallopian tube, this eggs travel or pushes further because of the help of cilia. They are more in number. You can see there are so many numbers as compared to flagella. Okay, cilia, they are so many in numbers, but flagella are few in number. They are few number. So let's talk about flagella. They are less in number. Undulating motion. Motion means what? Tail of a fish. Okay, a fish, how it do the motion? Okay, so it motion is what? That direction, in that direction. Okay, right, then again left, like this one. So that kind of motion is done by flagella. It's like a swimming one. Okay, it's like a swimming. So in that swimming also we used to do motion, undulating motion. This kind of motion is done by the flagella. So basically it also actually main role is swimming. Okay, it help in swimming, right, or doing the motions. Its structure is 9+2. I'll let you know about this one. What is 9+2? Again, it is assembly by the basal body like central. That also I'll let you know later on. Okay, let's talk about its structure. Okay, so see, this is a cilia. You can see, can you see this one is structure? It is the cilia or flagella. Okay, both. Okay, now if you remember from the cell, flagella are coming out like this one. Cilia are also coming out. The part of flagella which is inside this cell membrane, by which it just attach with the cell membrane, it is the basal body. So see, this is the membrane. Here this part of cilia which is inserted into this membrane, it is called basal body. Got it? Okay, yeah. So if we'll see the structure of this basal body, so here is like what? There are three microtubules are present. 3 microtubules are present. Okay, as I told you, triplet of microtubules at 9 places. Again, at 9 places, and nothing is inside. So it's again 9+0 arrangement. I can say 9+0 arrangement. In center there is no microtubule. 9 sets of triplets of microtubules are present over here. So this is the basal body. Okay, it will see the basal body of any cilia and flagella. Now the upper one body, this whole upper one body, and if we'll have a look of this upper one body, what is happen? I mean, what it has only 2 microtubule. So there is a 9 sets of doublet of microtubule. It's what? Outer microtubule which is doublet, and there are 9 in number. So there are 9 sets of doublet of microtubule, but here is triplet of microtubule. You can see here is triplet, here is a doublet, and in center also 2 microtubules are present. So that's why I'm saying it 9 plus 2. 9 doublets of microtubule and 2 microtubules are present in the center. Got it? What I was talking. So this is actually structure of cilia flagella. Here you can see this one is structure. Okay, and if we'll see the basal body of cilia and flagella, it is this one structure. So let me tell you again the further structure of cilia and flagella. So this is the basal body which is inserted. So if we'll see the basal body structure, it is 9 plus 0 arrangement. It is 9 plus 0 arrangement. And if we'll see the upper one part, here this one arrangement where 9 microtubules, doublets of microtubules, and 2 microtubule in the center. It is 9 plus 2 arrangement. Okay. Okay, so that's why this one line is there. Okay, so now can you understand now structure 9 plus 2? Can you understand this one?
Yeah.
And second is what? Assembly by basal body like centriole. So what it is? Assembly like they have the basal body which is exactly like centriole, because in centriole also I have told you this one is structure. So you can say basically cilia and flagella, they emerges from the centriole, or they form from the centriole, because the basal body is exactly like of centriole. So you can say that cilia and flagella emerges from centriole. Okay, because its basal body, it's exactly similar to centriole. So now let's talk about the detailed structure of this one, cilia and flagella. So see, now focus on this one structure. Cilia and flagella, if we'll see the cross section of cilia and flagella, there is the outermost layer. It is called the plasma membrane. It is called the plasma membrane. Now inside this plasma membrane, there is a doublet. There is a doublet of microtubule at 9 places. Okay, so you can see it is outer doublet microtubule. Outer doublet microtubule. Got it? Okay, now in inside center 2 microtubules are there. They are called the central microtubule. Okay, now all the doublets of microtubules, in which one tubule is called the A microtubule, second tubule is called the B tubule. Okay, A and so it is like that, A B tubule, like this one they are present. Okay, now the two center one microtubule are enclosed by a sheath which is called the inner sheath, and from each microtubule, from each A1 microtubule, there is a one structure arises which help them to connect with the center. Can you see this red one structure? Okay, through this it is connected to the center. All the microtubules are connected to the center, and this structure is called radial spoke. So how many radial spoke are there? It's 9, because 9 microtubules are there. So that's why it is 9 radial spoke. Got it? Okay, one more thing. All the microtubules, they are also connected. See, this one microtubule is connected with this one with the help of a red line. Okay, can you see all the red lines through which they all are connected? This is called nexen. Okay. Okay.
Yeah.
Now from each one microtubule, okay, in a doublet, from one microtubule, one more structure arising like this one. Can you see? It's like a feet. It's like a 2 feet arising from each one, and this is called the dynein arm. You have to remember this one. It is important. Okay, we'll talk about it later. So it is called dynein arm. Okay, so from each microtubule, 2 dynein arms are arising. Now there's a one function of it. It is bending. It is bending due to motor protein dynein. Okay, see, it is a protein which has 2 feet. I told you just now, na? These 2 feet. Okay, these 2 are what? Feet, right? That walk along the microtubule of adjacent doublet by, see, always remember, motor protein having the ATP. They utilize the ATP. Okay, they always utilize the ATP. ATP is what? Energy molecule. Energy molecule. So it utilizes the energy for, simply, if we walk, definitely we also need energy. The same way, if motor protein is used to walk, they also need energy, and they need energy, so they utilize the ATP to do its function. So they have also 2 feet like we have. They also walk like this. Suppose if we walk, when we walk, what we do, our 1 feet is attached or touches the ground, and 2nd feet is above the ground. Okay, and when we comes or when we go forward, our 2nd feet again it touches the ground, and the first 1 feet it detaches from the ground. Okay, like this it also walk, right? So what is doing? One foot maintain the contact while the other releases and reattaches further along the microtubule. Like we walk, same way they also walk. Okay. So doublets and the 2 central microtubules are held together by a flexible cross-linking protein. This is what they are saying. This one. They are saying about this one. Okay. They are attached with the cross-linking protein. Movement of dynein feet help to bend. So this is why, because they dynein feet, they show the movement, and because of this they are able to bend. They are able to bend like this. Okay. Microfilament. Okay. I told you there is a 3 component in cytoskeleton. One is microtubule, second is microfilament, and the third one is intermediate filament. Okay, so second one is microfilament. Okay, yeah. So what is microfilament? I told you it is made up of actin filament. Remember, 2 filament, they coiled around each other. So it's a very solid rod and thinnest. Okay, twisted double chain of actin subunit. Twisted, it is twisted. Okay. Structural role. Now where is perform the role? First of all, it bear the tension. Okay, it support the cell shape. Make core of microvilli. Okay, so do you know the structure of microvilli? Okay, you must be knowing intestine. So what is actually intestine? It folded inside like this one. Okay, and this infolding is called the microvilli. Okay, what is the role? Because it enhances the absorption area, because in intestine maximum digestion takes place. All the nutrients are released inside the this intestine. So these microvilli help in absorption, maximum absorption of these nutrients, and give it to our body. Okay, so it make the core of microvilli. So if we'll see this microvilli, this is the microvilli, and if we'll see inside the structure of microvilli, it is basically made up of microfilament. Okay, the delicate projection that increases the cell surface area, it is actually microvilli. It is a delicate projection that increases the cell surface area. So these all are the structural role of microfilament. Now what is the basic function? It helps in cell motility. Actin filament, what it do? There's one more protein which is called myosin. Okay, it is a thicker filament. It is also a motor protein. Okay, like dynein, it is also a motor protein. Actin interact with this myosin and cause the contraction of our muscle cell. Okay, can you see our muscle cells, how our muscle cells work? If our muscle cells contract and relaxes, okay, this is how we are able to do the movement. If we are walking, if we are moving our hand, if we are talking, our muscles are working. So how muscles work? It contract then relax, contract and relax. So this contraction is because of the microfilament, because in microfilament this actin filament interact with the myosin, and basically this myosin, actin, they both are present in our muscles, so they interact and contract and relax. Okay, it also help in contraction in amoeba, WBC. Yeah, so if you remember, this is amoeba, right? I've told you. And whenever there's any food particle, so what it do the movement, or there's a contraction, relaxation in its membrane, and that's why it's able to form the pseudopodia. Pseudopodia is what? It is a false feet. Okay, here in WBC as well, it make the pseudopodia. Cytoplasmic streaming in plant cell. Now what is cytoplasmic streaming? You must be knowing a stream. What is a stream? Okay, the where the liquid, or you can say water, it just move on, that is called a stream. Okay, but if the cytoplasm is move on, it just move in the way of a stream, then it is called cytoplasmic streaming in plant cell. So in a plant cell, the whole cytoplasm is move, it's move. Okay, that is called cytoplasmic streaming. So it is also because of the microfilament. So did you get it? What are the roles of microfilament? Third component, it is intermediate filament. Okay, as I told you, it is of intermediate diameter, not more less thicker, but more than thinner. Okay, so it is intermediate diameter. It is formed from molecular subunit of protein, which is keratin protein. It is a protein. Keratin is present basically in your hair, in our nails. That is strength. Okay, that is strength is because of the this keratin, and keratin is a part of intermediate filament. So keratin is a protein. Okay, so in protein there are certain molecules. From that molecule it is formed. Okay, so it is formed from the molecular subunit of protein, that is keratin. It is due permanent fixes of the cells. Cells are fixed. Okay, cells are fixed because of the this intermediate filament. Okay, one more thing, remember, intermediate filament is used to denote by IF. Okay, intermediate filament. Now one more thing, because it is a very hard, I mean, like, it is always there, it do not destroyed and all that. So if an organism is die, IF, that is intermediate filament, its network still persists. Okay, it is still persist. It reinforcing the shape of the cell, it maintains the shape of the cell and fixing the position of certain organelles. Okay, so basically it is a fixer. It is a fixer which fixes the cell at its place, and even the cell die, it never ever, it also never ever just vanish, it persists. Okay, like for example, nucleus sits in a cage made of IF. Okay, let me show you. Suppose this is a cell and nucleus is present over here. It is nucleus. Okay, so nucleus in its position and it's in shape because of this intermediate filament. So intermediate filament is present outside this one. Okay, like this one, like this one. Okay, so it make a cage like structure in which this nucleus fit or sits. This is how it fixes the cell. Okay, so nucleus will not move from one place to another, it is only there own. Okay, as well as the nuclear lamina. So you must be remembering nuclear lamina, this is the nucleus. Okay, double membrane structure. Inside this inner membrane, this is called nuclear lamina. It is also IF, made up of IF. So see, outside one is also made up of IF, inside. So this is how the nucleus membrane, it is fixes at its place and maintain the shape of the organelle. Got it? So there's a one question, how cilia and flagella bend.